Okay, we are recording now, so I want to finish talking about kingdom Animalia, the animals. Animals, if you recall, are multicellular with cell differentiation, which just means they're made up of many cells, and each animal body has not just many cells, but many cell types. The simplest might have maybe 10. You've got about 200 different types of cell. Animals are generally heterotrophic, Greek for others feeding. They have to eat other living things, plants or animals. What I mean is they have to eat animals or they have to eat plants. Most of them can move at some stage in their life cycles. We say that they are motile. They're usually symmetrical, meaning that there is at least one way that you could divide an animal body into mirror image halves. For things like sea urchins and starfish, there may be more than one way, but usually there is at least one. They don't have cell walls. Cells are held together by a fiber meshwork called the extracellular matrix. This is what enables your tissues to be flexible. The reason why most of your body is not wooden in texture. And all but the very simplest organisms have cells organized into tissues, which are coordinated groupings that are specialized for one function. And in animal bodies, you've got four basic tissue types. Epithelial tissue is tissue that lines cavities. So the inside of your mouth is lined with epithelial tissue. The inside of your nose is lined with epithelial tissue. The inside of all your digestive tract and your bladder and whichever set of reproductive bits you happen to have, and your lungs and your windpipe and your blood vessels, all of those cavities are lined with epithelium. Incidentally, I should also have included your skin is made of epithelial tissue. That lines your outside. Epithelial tissue is often modified to secrete. So your salivary glands, the glands in your mouth that make your saliva, are made of modified epithelial tissue. The sweat glands in your skin, the mucus glands in your lungs, and the enzyme glands in the walls of your stomach, those are all epithelial tissue. Connective tissue is generally used in structural support. Your bone is made of connective tissue. Your bone is not dead. It's very much a living tissue. I am told that orthopedic surgery is some of the bloodiest that there is because bone has lots of blood vessels in it. It's very much alive. Your cartilage is a connective tissue. Your tendons are connective tissue. The Achilles tendon that you can feel at the back of your heel, connecting your calf muscle to your heel bone, that's connective tissue. The big sheet-like tendon at the back of your neck that connects the back of your skull to your trapezius muscles, that's connective tissue. Blood is technically considered a connective tissue, even though it's not really used for that kind of structural support. But most connective tissues lock things together, hold things in place. The loose, flexible, springy stuff under the skin of your face is also a connective tissue. And then the other two you probably already know. Nervous tissue is specialized to transmit signals. Your brain is mostly made of nervous tissue. So is your spinal cord. So are your nerves. And then finally, muscle tissue is tissue that contracts and is used in motion. So your muscles are mostly made of muscle tissue. So is part of your So are some layers that make up your stomach and intestine, for example, because those have to contract in order to churn up your food and digest it and squeeze it on through. There's certainly your heart is largely made up of muscle. You've got muscles in the walls of your blood vessels. That's why some people turn pale when it's cold and flushed when it's hot, is that those blood vessels can close to tighten up the blood supply to the skin, or open it up and let lots of blood through the blood vessels in the skin. And that's what changes your skin tone from red if you're hot or if you're blushing, to more pale if you are if you're cold or if you're frightened or something like that. So those are the big four. Just a couple of examples here. This is a slice through the lining of a human fallopian tube. You're going to see epithelial cells in lab taken from your own skin. And those are going to look like flat, slightly crumpled, a little bit like crumply tiles, because they're very flattened and they fit together edge to edge, kind of like tiles on a floor. The epithelium in your fallopian tubes, if you're one of the people that's got those, the cells aren't shaped that way. This is called columnar epithelium because the cells are very long and narrow and they're packed together like a bunch of columns. So I'm drawing in a couple, and you being eukaryotes, each one of these has a nucleus. So there are several different types of epithelium in all of your passages. This is what cartilage looks like under the microscope. The actual cells are these white things, or they were in those white things. Some of them seem to have dropped out, but that's where the cells would go. And the pink stuff surrounding the cells, the stuff that's stained pink, remember, this is a stain. Your cartilage is not really pink. But all of the stuff that separates the cells, that is very tough variety of extracellular matrix. Tough and rubbery type. And cartilage, of course, is what supports your ears and your nose. And an even tougher variety of cartilage is what makes the surfaces of your joints. And if that cartilage wears away, you have one particularly bad form of arthritis. So there, that's an example of connective tissue. That's what nervous tissue looks like. That is a piece of human brain that has been sliced and stained and put under the microscope. Those are brain cells. Notice they've got lots of projections coming away from them. Adjacent nerve cells connect to each other through those projections. Those would be, the thicker ones would be called axons, and the thin ones dendrites. We will not really have time to get into how brain cells connect with each other and what kind of signals they send. There's just not time to cover all of that. All we need to know for our purpose is that nervous tissue is involved in signaling. And then finally, that's a piece of human skeletal muscle. The muscle cells are very long and fibrous, like that's why meat has a fibrous texture. It's made of muscle cells that are all organized in one direction. That's why meat has a grain. And the nuclei are right here. And each muscle cell, at least in skeletal muscle, each muscle cell has several nuclei, because sometimes that happens. But most of the cell is made up with this stuff that stains pink. And if you look very carefully, you can see it's got a striped appearance. We're not going to have time to go into why, but let's just say that striped appearance is due to the presence of a lot of specialized proteins that enable the muscle cells to get shorter, enable the muscle to contract. Incidentally, muscles get shorter, but they can't get longer on their own. For a muscle to extend, another muscle has to stretch. So if you contract your biceps, you flex your elbow. To extend your elbow, your biceps can't push. Your biceps can't get longer on its own. You have to relax your biceps and contract a muscle on the opposite side of your arm, your triceps. So muscles tend to come in these opposing pairs, where each one can contract and then be stretched by the contraction of the other muscle. In some of the simplest true animals, ones that we put in the phylum Cnidaria, the body is made up of several types of tissue. There is, in a jellyfish, one type of epidermal tissue on the outside and a different type that lines the stomach and so on. And there's nervous tissue in between the two. And in jellyfish and corals, that's pretty much all there is to it. But in most animals, you can get several tissues coming together to form an integrated unit called an organ. This just happens to be a slice through the wall of your small intestine. This was an organ that I kind of picked at random. What's lining the inside right here, all of this that I am aligning, all of that is epithelial tissue. A lot of it is modified for secretion. The lining of your intestine secretes mucus, for example, to lubricate the passage of digested food. It's also modified for absorption. This is where nutrients from your food get taken into the body bloodstream. So there's epithelial up there. There's another layer of epithelial on the outside, which I'm aligning. Below the epithelium, we've got thick muscle layers. That's one layer of muscle that when it contracts makes the intestine, let me think, makes the intestine narrow and long. No, sorry. It's this layer below it that makes the intestine get longer and narrow. The layer on top makes the intestine get shorter and thicker. And working opposed to each other, they contract in waves that push digested food down your intestine from one end to the other. So a lot of the lining of your intestine is muscle. You've got epithelial on the outside. There's connective tissue in here and connective tissue in here, holding the whole thing together. And you don't see them in this picture, but there also are nerves in your intestines that are involved in controlling digestion. They can slow down or speed up the rate at which food passes through, for example. So your small intestine has all four types of tissue integrated into this unit. And then when you put several organs together that are integrated together, that makes up an organ system. This is the human digestive system, of course. Human anatomists generally recognize about 15 others. Some of you may go on from here, if you're going into the health sciences, to take anatomy and physiology or structure and function of the human body. You get to learn all about the organs and organ systems there. But this would generally be true of all animals. Even a cockroach, for example, even a fly has organs and organ systems. Pretty much all animals except for jellyfish and corals and sponges we could talk about in this way. Okay, all right. How's everybody doing? All right. That's all the living biodiversity that we have time for. But one extremely important discovery made by many people over the course of a couple hundred years is that living biological diversity is not all there is to the story. There is dead biodiversity as well. Things like this Tyrannosaurus Rex on display in Chicago, I believe. I think that's at the Field Museum of Natural History. Even in prehistoric times, people would occasionally dig up rocks that had funny little shapes in them. Sometimes the rocks would look like bones. Sometimes the rocks would look like shells or teeth, or sometimes even plants. And people have always wondered what they were. The ancient Greeks are thought to have taken some inspiration for some of their monsters by looking at certain fossils that you can find to this day on some of the Greek islands. What we've worked out is that what we now call a fossil is any trace of a once living organism, a living thing that used to be alive and now is not. We can talk about body fossils, that thing that just skated in is a rock dug up just outside the town of Kemmerer, Wyoming, probably in Fossil Butte National Monument, a place you can visit if you're ever out that way. And it is pretty clearly the skeleton and even some impressions of the soft parts of a fish. In fact, it's pretty similar to modern day brim or bluegills or sunfish or Sinia jabrania solnichniki, or whatever we're calling them today. It's actually called Priscacara pili, if memory serves. But that's clearly an animal's body. Most body fossils don't look nearly that nice. Most fish fossils are just scattered bones. This just happens to come from a place where fish skeletons are preserved in incredible detail, so much so that it's protected as a U.S. national monument. Down below it, that's a picture that I took in far northeastern New Mexico at a place called Clayton Lake State Park. There's a nice lake there. It's an artificial lake. They built a dam to create it. And if you hike back a ways to where they took the earth to build the dam, they exposed a lot of rock layers below. And those rock layers contain impressions, dents in them, that look like that. I'm outlining one of them in red right now. There's another one up ahead, right about there. That's the other reason you might want to go to Clayton Lake State Park someday. That is a part of a dinosaur trackway. And this is an example of what we call a trace fossil. It's not an actual body, but it's a trace of a living thing's activity. That's there because a long, long time ago, roughly, I think about 90 million years ago, although I'd have to look up exactly how long, that rock used to be mud, and a dinosaur walked across it. In fact, a bunch of them did. There are lots of tracks that you can see at Clayton Lake State Park, although they haven't been taking good care of them, so some of them may have eroded away. But at least when I was there, you could still see quite a lot of them. We get this in Arkansas. The largest dinosaur trackway ever found was actually excavated in a gypsum quarry just outside the town of Nashville, if anybody knows where that is, in the southwest part of the state. Unfortunately, the quarry owners didn't preserve it all because they would have basically had to shut down. But we did take some rubber molds of those tracks. And at least a few years ago, some of them were on display in the Museum of Discovery. So tracks like that are not uncommon at all. And that's a second type of fossil, a trace fossil. Virtually all the rocks, with a few odd exceptions, let them go. Virtually all rocks where you can find fossils are what we call sedimentary rocks, meaning that they were formed by water or occasionally by air, laying down layers of mud or silt or sand. That's the way that most of the rocks that you see here in the wall of the Grand Canyon formed. Some sedimentary rocks were laid down when a body of water evaporated, like the layers of rock salt deep under South Arkansas, or they were laid down through various chemical reactions, like some types of limestone, or flint is another one that's laid down chemically. And there are other types of rock, of course. There's igneous rock that used to be molten, and there's metamorphic rock, which is formed by heat and pressure acting on different rock types. But almost all of the rocks that you're going to find fossils in are sedimentary rocks, laid down by the action of a fluid, usually water, occasionally air, which counts as a fluid because it flows. So how'd they get there? The answer was worked out by a guy named Nicholas Steno, died in 1686. He was trained as a physician. He lived in Denmark. He actually, in the middle of his life, ended up giving up his medical practice, becoming a Roman Catholic priest, ministering to the Catholic minority in Denmark, because this was a period in history when people were killing each other over whether they were Catholic or Protestants. This was not an easy time to be Catholic in a Protestant country. And Steno was actually beatified in 1987. So he is recognized for his holiness by the Roman Catholic Church. But aside from that, he was one of the first people to try to think in a rigorous way about how you got fossils, how you got bones of what looks almost exactly like a fish embedded in rocks in the middle of, the Wyoming desert, where there certainly aren't any fish today. How do these things get into rock layers? Well, he thought and thought about it and observed rock layers around where he lived and came up with a couple of principles that we now call Steno's laws. The first is Steno's law of superposition, and it tells you this: rock layers are laid down, sedimentary rock layers are laid down in layers. And anytime you've got a stack of sedimentary rocks like these, the Vermilion Cliffs in southern Utah, the rocks on the bottom of the stack are the oldest that you see, and the rocks on top of the stack are the youngest that you see. And it's fairly easy to demonstrate this. Fill a bucket of water sometime and throw in a shovel of gravel. Let it settle, and then dump in a shovel full of mud. Let that settle out, and then dump in a shovel full of sand, let's say. What you'll end up with is 3 layers. You'll have the gravel on the bottom, the mud in the middle, and the sand on top. And the sand is on top because it was the last layer to be dumped, right? So the only place it can go is on top of the layers that formed earlier. Right, the gravel and then the mud. So in that bucket, the oldest rock layers are on the bottom, that's the gravel. The youngest rock layers are on top, that's the sand, and the mud is in the middle. Sounds really obvious, but it wasn't until Steno got around to actually stating it. The exception is if something disturbs rock layers after they form. These have been thrust upwards to make the cliffs, and they've been eroded, but they haven't really been messed with, and the order of appearance is still intact. Steno's law does not say anything at all about how old rock layers are. I've hiked in my younger days over the layers on the left. They're in a place called the Drum Mountains in western Utah. And you can see these rock layers up here, and the youngest are up at the top, and the oldest are these down at the bottom, and the middle are in the middle. Right, so we can go from oldest layers to youngest layers by climbing up that way. We can travel in time, as it were. Now, where we get these numbers, I'll try to talk a little bit about later. Don't worry about it for the moment, but I will get to where we're getting these dates. The layers on the left formed approximately 500 million years ago next Thursday, and it took several million years for those layers to form. And incidentally, they formed in the ocean, and a lot of geology has happened to put those layers in the middle of what's now the Utah desert and thrust them up to form a set of mountains. Okay, that's a very long story and more than I've got time for, but that's what happened. On the right, that's a picture I took at a place in Death Valley National Park that you ought to go visit sometime, called Ubehebe Crater. And there are rock layers right here on the rim of the crater. And they also go from oldest down here at the bottom to the youngest up there at the top. And it's hard to see the layering. You may have to look pretty carefully, but there is black and gray striping on those rocks. They are, in fact, divided into layers. The rocks on the rim of Ubehebe Crater formed a few thousand years ago. They formed, in fact, when this volcano erupted. There would have been Native Americans in the area who might have seen it happen. So there were fully modern people here. This was not that long ago. This is maybe about the time the Egyptians were building the pyramids, roughly. And we think those layers were laid down possibly over a period of just a few weeks. Steno's law does not say anything about the rocks having to be exceptionally old, and it doesn't tell you anything about how old they are or how long it took them to form. All it is a statement of relative age. In both of these pictures, the layers on the bottom of the stack are older, and the layers on the top of the stack are the youngest. And if you climb that stack, you go from the oldest layers to progressively younger and younger layers, regardless of whether younger means 10 million years younger or a week younger. And rock layers can be tipped and folded in various ways after they form. Quite a lot of rock folding associated with the movement of continents, which is not something we've got time for here, I'm afraid, is what made the Ouachita Mountains. These are rock layers that are exposed just south of Hot Springs, somewhere on McLeod Street. And you can see these layers, instead of being horizontal, are crumpled like a beer can against my grandmother's forehead. You can see how they make these folds. That's, the Ouachitas are basically this giant crumple zone where rock layers were slammed together because of the motion of continental drift and crumpled up and thrust up into mountains, similar to what is going on now in the Himalayas, which are forming in the same way. You can see this you don't get bends quite like this in Conway, but if you ever drive out Hogan Road to or from US Highway 64, Hogan Road goes right through Cadron Ridge, and there is a big road cut right there. And there are a lot of rock layers that you can see that are parallel, but they're tipped on their edge, angled about 55 degrees. That's part of this same fold belt. The bedrock under Conway is sedimentary rock, but most of it has been folded and tipped like these rocks have. This sometimes can mess up the application of Steno's law, but usually you can work out what rocks are the oldest and what rocks are the youngest. There aren't that many cases where you really, in practice, have a hard time telling. Enter Will Smith. Not the Will Smith who started out as the Fresh Prince of Bel-Air and made those records with DJ Jazzy Jeff, and is apparently starring in a really good movie about Venus and Serena Williams. This William Smith was English. He was a surveyor, and he happened to work at the time of the Industrial Revolution, when Britain was industrializing, building factories, and faced with the problem of how to get materials from, the mines to the factories, and how to get factory product from the factories to the places where it needed to go. A little bit before railroads, they built a system of canals. So you could ship things from one end of England to the other almost on barges in these canals. Smith was involved in surveying canal routes, so he saw an awful lot of rocks blasted through by the canal diggers, and most of that rock is sedimentary, and Smith saw a lot of rock layers and picked up an awful lot of fossils, built up a huge collection. And he realized, first of all, different rock layers contained different fossils. You could find, I don't remember if he found dinosaur teeth, but you could find, let's say, what the heck, dino teeth in some rock layers, but you didn't find them just everywhere. And what was more, fossils occurred in a predictable order of appearance. So if you found dinosaur teeth in one rock layer and, let's say, oyster shells in a rock layer above it at one place, that meant that at that place, dinosaur teeth were older and oyster shells were younger, because that's Steno's law. And then he found that everywhere you went, if you found those two fossils, you would find dinosaur teeth before in older rock layers than the ones where you found oyster shells. And that's called Smith's law of succession. And here's how it works. These are rock layers that I have hiked over many a time, and I've confirmed a lot of this with my own personal observations. Anybody who wants to spend a lot of time hiking the Spring Mountains outside Las Vegas can see what I have seen. On the left, that's a diagram of rock layers. At the bottom of the stack that I've drawn is a layer of some pretty thick rocks called the Wood Canyon Formation. On top of that, so younger, is a layer called the Zabriskie Quartzite. On top of that is a layer that's mostly very thin shales called the Carrara Formation. And on top of that, so the youngest of these four, is a rock layer of very heavy and sharp magnesium-rich limestone that's called the Bonanza King Dolomite. By the way, rock layers are named after places. The Bonanza King was the name of a mine. And the first scientists to name these rock layers must have been working close to the old Bonanza King mine. And Wood Canyon's a canyon, and Zabriskie and Carrara are now ghost towns. Okay, mute your mic, somebody. Sounds like you're zipping up things. Thanks. Okay, if you go poking around in these rocks, as I have, you will find the fossils of critters called trilobites. Trilobites are extinct. We don't have these anymore. If you saw one, you might think it looked like a roly-poly or an insect or something like that. It's got a segmented body, and it would have had lots and lots of legs. If you go poking around in the bottom of the Wood Canyon Formation, you can find trilobites that belong to the genus Nevadia. Hike up section a little bit, as we say, and go digging in the bottom of the Carrara Formation, and you can find plenty of trilobites that belong to the genus Olanellus. And Olanellus does look pretty similar to Nevadia, but there are differences, and with a little practice, you could learn to spot them pretty easily. It's not something you need a PhD degree to be able to do. There's a lot of amateur fossil collectors that have learned. And then if you go higher in the Carrara Formation, so into younger rocks and dig, you won't find Olanellus, you'll find trilobites in the genus Cispacephalus, which you can see looks quite different from either Nevadia or Olanellus. Okay, so what? Well, if you go to, you don't find these trilobites everywhere in the world. You won't find any of them in Arkansas, for example. But if you go to places like British Columbia or the Northwest Territories of Canada, and you dig in the right rocks and you find these trilobites, you'll find them in this order. Nevadia is the oldest, Olanellus comes after, Cispacephalus is the youngest. You'd find the same thing if you dug in parts of Newfoundland on the Atlantic coast of Canada. You could find the same thing in certain parts of North Georgia. You could find the same thing if you're in the right regions of Siberia. You could find the same thing if there's parts of not Africa. Okay, there's parts of Scandinavia where you could do the same thing. If you find these fossils anywhere, you find them in this order of appearance. It's the same all over North America and actually over a pretty good sized part of the world. So put this observation together with Steno's law, and we can say that Nevadia lived first, and then it seemingly went extinct and was replaced by Olanellus, which lived for a long time after Nevadia. And then when Olanellus died, Cispacephalus appeared and was the youngest of the three. You see how the logic works behind this. I haven't said anything about how old these guys are or how long they survived, but we can tell that one of these lived in a time, in the oldest time, that's Nevadia. Olanellus came later, and Cispacephalus is the latest of the three. Where they came from, interesting question, and we'll get to that on Wednesday. But for right now, just know that this is the order in which they appear. And you can do this for all kinds of fossils. In fact, oil companies hire a lot of fossil specialists, a lot of paleontologists, who use the order of appearance of fossils as a tool for determining how far down a well has been drilled and what rock layer they're in, and whether they might need to drill farther or whether they've drilled too far. Yeah, oil companies hire paleontologists and use this knowledge of the order of appearance of fossils. They use that to determine where their drill is. You put gas in your tank, thanks to the work in part of quite a lot of fossil specialists. The fossil guy who figured out how to do this was one of the few who ever died wealthy. And what this order of appearance of fossils lets us do is define time periods in Earth history based on what was alive at different times. So there was some time in the past when the dominant land animals were great big dinosaurs, and also very small dinosaurs as well. Dinos had lots of different sizes. And then at some time after that, the dinosaurs went extinct and were replaced by large mammals. So we could talk, if we wanted to, about the age of dinosaurs, which eventually came to an end, and the age of mammals began. And we can break down Earth history, because Earth has a history, and we can break it down into time periods, just the way we could break down European history into the medieval period and the Renaissance and the Protestant Reformation and the Industrial Revolution and the Age of Empires. Just as we could break down history into blocks of time, we can break down Earth's history into blocks of time. And by about 1830, just about all scientists who had studied the matter agreed that there had been lots of different types of living things on the Earth. Many of them had gone extinct. These were some of the first attempts to reconstruct what dinosaurs looked like. We now know they're not very accurate. And it was accepted by just about everybody that the Earth was really old. Exactly how old, nobody knew, but indefinitely old, and that the Earth had a long history. And you cannot fully understand life today unless you go to places like Dinosaur National Monument outside of Vernal, Utah, where this picture was taken, and study something of the history of life. Just as you can't understand contemporary politics without understanding history, just as you can't understand America without understanding American history, the good and the bad, you can't fully understand life without knowing something about life's history, because life has a long history. And on that note, I will stop the recording. And hang on. All right, sorry, I'm going to stop the recording.